Good day all. My name is Dr. Ifeanyi Mary Ann Okafor. Thank you very much for being part of Hematology and Blood Transfusion Science lecture series. Last time we started lecture on iron metabolism, and today we are going to continue with that topic, but we are going to be dwelling on iron absorption today. Feel free to subscribe if you have not subscribed. Permit me to share my slides with you. We will be discussing iron absorption today. And the objective of this lecture is expected that at the end of this lecture, you should be able to mention key proteins that are associated with iron absorption, describe iron absorption and the fate of absorbed iron, understand the mechanism of transport of iron across the intestinal mucosa, and should be able to mention factors that can influence dietary iron absorption. In our previous lecture, we talked about iron being essential mineral that is required by every living cell for so many metabolic processes, be it DNA synthesis, oxygen transport, respiration, energy. And iron has a very important property that helps it to achieve all these important functions. The ability of iron to shuttle between two oxidative processes, the ferrous ion and the ferric ion. And this is capacity of iron to move from ferrous to ferric ion and from ferric ion to ferrous ion within the same system is actually what helps it to achieve its clinical enzymatic functions or be part of most of the critical enzymatic reactions, especially the one that concerns with oxygen transport, electron transport, and cellular energy production. I said that to balance iron homeostasis, to balance iron metabolism, the individual, the balance predominantly reflects these variables. That is the nutritional intake, the amount of iron that you take in and absorb, and then the iron loss, the amount of iron that is recycled in the body as part of catabolism of red blood cells, and then the current demand of an individual, the physiologic state of an individual helps us to achieve an iron. The interplay between these variables helps to achieve what's called iron balance in an individual. Then we said that we have two forms of iron in our diet, the heme iron and the non-heme iron. The heme iron and the non-heme iron. And we say that the heme iron we can get from meat as part of hemoglobin and myoglobin, from meat, poultry sources, and fish. And then we have the non-heme iron we get from cereals, grains, legumes, fruits, and certain vegetables. And for iron absorption proper, heme iron and non-heme iron somehow are absorbed differently, even though at the end of the day, they enter into the same common pool. Iron absorption. Iron normally enters the body through gastrointestinal tract, the GIT, and it is mostly through the proximal duodenum. Iron enters into the system through the proximal duodenum. That is, when you take in iron through your food, goes through your GIT and straight down stomach and to your proximal duodenum where it is absorbed. The point of absorption of iron in our system is at the proximal duodenum. The amount of iron absorbed is normally tightly regulated according to body needs. Just like I said earlier when we are looking at variables that helps in iron homeostasis, the nutritional intake, that is the iron that is absorbed, the iron loss, the current demand, that is physiologic state of the individual, and then the amount of iron that is recycled in the system, that is the internal supply of iron. So the amount of iron that is absorbed is normally tightly regulated according to body needs. So a pregnant woman is expected to absorb more iron than a non-pregnant woman. And then infants and somebody in anemic state is expected to take in more iron than somebody that is not in anemic state. Now for active erythropoiesis, iron deficiency upregulates iron absorption, while iron overload downregulates iron absorption. Mechanism of transport across the intestinal mucosa. How is iron transported across the intestinal mucosa? The pathways for the absorption of inorganic iron. Inorganic iron is iron, non-heme iron. Non-heme iron can also be called inorganic iron. Remember I said our diet contains two forms of iron, heme iron and non-heme iron. The pathway for the absorption of non-heme iron and heme iron are somehow different. These pathways do seem to merge within the intestinal cells, even though at the luminal side, they might absorption of non-heme iron and heme iron might seem a little bit different. But these two pathways seem to merge within the intestinal walls. Across the intestinal walls, it might seem different, but once within the intestinal walls, the two pathways seem to merge, but the whole iron is put inside a common pool. The entry of iron inside the intestinal cells, research has been going on in the past. Many researchers have done work, around this, and have found out that the existence of a pathway in which beta integrin and the protein designated as morbilferrin are involved in transporting iron. These proteins are believed to form a complex that has been designated paraferritin. They form a complex that has been designated paraferritin, even though they may not contain ferritin. And this complex contains an important ion transporter that is called divalent metal transporter 1. Divalent metal transporter 1. Also, paraferritin has ferrireductase activities. Paraferritin has ferrireductase activities. And then duodenal cytochrome B reductases reduces ferric ion to ferrous ion. Duodenal cytochrome B reductases reduces ferric ion to ferrous ion. Iron is reduced by duodenal cytochrome B iron is reduced by duodenal cytochrome B, and then the ferrous ion is then transported into the cell by means of divalent metal transporter 1. Divalent metal transporter 1. To exit into the plasma from the luminal surface of the cell, the ion is reoxidized by histine and transported from the cell by ferroportin. We are going to look into this in detail in a few seconds. The luminal non-heme ion, that is the luminal non-heme, the ion that we take in our diet occurs in form of iron 3, that is ferric ion. And this iron in our diet must be reduced first to iron 2. before they can be absorbed. The iron in our diet, that is non-heme iron in our diet, most times occur in form of ferric iron. And for them to be absorbed, they must be reduced to ferrous iron, that is iron 2. And we have enzymes on the luminal surface of the enterocytes, duodenal enterocytes, that helps in reducing this iron 3 found in our diet to iron 2, so they can be absorbed across the luminal wall. These ferroreductases, such as cytochrome B and STEP3, are found along the luminal wall of the duodenal enterocytes, and they help in reducing iron 3 found in our diet to iron 2 before they can be absorbed across the luminal wall into the intestinal cells. Now, iron 2 is then transported once it's been reduced from iron 3 to iron 2. This iron 2 is then transported across the apical membrane by divalent metal transporter 1. Another name for divalent metal transporter 1 is natural resistant associated macrophage protein 2. Natural resistant associated macrophage protein 2. But for the purpose of this lecture, we are going to stick to divalent metal transporter 1. So where the non-heme iron in our diet occur in form of iron 3 and must be reduced to iron 2 before they can be absorbed across the luminal wall. And the enzymes that help in reducing this iron 3 to iron 2 are called ferroreductases, such as cytochrome B or STEP3 enzyme. The absorption of non-heme iron is inhibited by substances in diet that bind and stabilizes iron 3, while they are enhanced by substances in our diet that stabilizes iron 2. Heme iron is carried by the non-heme iron is reduced from iron 3 to iron 2 before they can be absorbed across the luminal wall by divalent metal transporter 1. While the heme ion are carried across the luminal wall by heme carrier protein 1. Heme carrier protein 1. They are carried across the luminal wall into the duodenal cells by heme carrier protein 1. But inside the intestinal cells, it is metabolized. The heme ion is metabolized into iron 2, which enters into a common pool with non-heme ion. They enter into a common pool with non-heme ion. Let's look at diagrammatic explanation of what I have just described. You look at this diagram, we have the food ion. We have two forms of iron in our food, the heme ion and the non-heme ion. The first one there, the heme ion, is carried across the luminal wall. Remember I said that the ion is absorbed at the point of proximal duodenum. So at the luminal side of proximal duodenum, the duodenal enterocyte, the heme ion is carried across the luminal cells into the intestinal cells by heme carrier protein 1, that is the heme transporter. Why the non-heme ion that we eat, and like I said, the non-heme ion and iron that are found in cereals, grains, legumes, vegetables as part of the food we eat. Why the heme ion are iron that are found as meat, fish, and poultry sources like egg, chicken, But this heme ion moves across the luminal wall directly by heme transporter, heme carrier protein 1. Why the non-heme ion that are in our diet are there in form of iron 3, and for them to be absorbed, they must first of all be reduced from iron 3 to iron 2. And we have duodenal cytochromes, enzymes that are formed along the duodenal walls that help in reducing this iron 3 to iron 2. These duodenal cytochromes are like the duodenal enzymes are cytochrome B. Cytochrome B, that is ferroreductases. Cytochrome B, they help in reducing non-heme iron that are in form of iron 3 to iron 2 for them to be absorbed. And then when it is reduced to iron 2, then you will have a carrier, a transporter called divalent metal transporter 1 that now carry the iron 2 across the luminal wall into the intestinal walls, intestinal cells. When that is done, the iron 2 are moved into the cells, and two things happen. When iron enters the duodenal cells, they can follow one of the two pathways. They can they can be transported to the blood and incorporated into ferritin, or they can be stored as mucosal iron in form of ferritin on the mucosal wall. You can see here, they can be stored as ferritin, mucosal ferritin, or they can move as iron 2, and then the heme iron that has been moved into the duodenal cells move into a common pool as iron 2, and then they can now move across the iron 2, iron destined for the circulation. If they are not stored on the mucosal cells as mucosal ferritin, the one that destined for the circulation is now transported from the cytoplasm across the basolateral enterocyte membrane by what we call ferroportin, by a channel called ferroportin. This process is coupled to the oxidation of iron 2 iron to iron 3, which is carried out by the iron oxidases, heptene, ceruloplasmin, and cyclopen. The newly absorbed iron 3 ion binds rapidly to the plasma protein transferrin, which delivers iron to red cell progenitors in the bone marrow. Let us look at our diagram again. When iron, remember I said, iron in our food occurs in two forms, as heme iron and non-heme iron. Heme iron basically is carried across the luminal wall. That is, iron is absorbed at the proximal duodenum. It's carried across the luminal wall by heme transporter. That is heme carrier protein 1. But non-heme iron occurs in our diet as iron 3 and must be reduced to iron 2 before they are absorbed. And duodenal wall contains enzymes that help in ferroreductases that help in reducing iron 3 to iron 2. And this ferroreductases is, for example, is cytochrome B. Cytochrome B helps in reducing the iron 3 found in our diet to iron 2 in the duodenum before they can be absorbed. So iron 2 is now absorbed through a carrier protein called divalent metal transporter 1. This divalent metal transporter is the channel through which iron is absorbed. Is the channel through which iron is absorbed. Divalent metal transporter 1 carries iron in form of iron 2 across the luminal wall into the duodenal cells. Inside the duodenal cells, two things happen. The iron from the heme iron and the iron from non-heme iron forms a common pool as iron 2 within the duodenal cells. And they can be stored in ferritin, mucosal ferritin, or it can be destined for circulation. The iron that is destined for circulation moves into the basolateral wall of the duodenal walls. There is a channel there called ferroportin. This channel is the channel that allows basolateral side of the duodenal walls to move iron into the circulation This channel allows iron to move across the duodenal wall, duodenal enterocyte into the circulation. Here, iron, you see the iron, absorbed iron in form of iron 2, moving through the ferroportin, to go enter into the circulation. But for this iron 2 to be incorporated into transport protein, remember we said iron cannot be moved unbound in plasma. Iron must be incorporated into proteins. And the protein that transports iron in plasma is transferrin. So when iron is absorbed and moved through the ferroportin and into the plasma, and iron moves across the ferroportin in form of iron 2, and this iron must be converted back to iron 3 before it can be incorporated into the plasma transferrin. And along the duodenal enterocytes, we have the enzymes hepstine, which converts the iron 2 back to iron 3. Remember, in the other luminal side, the iron is reduced from iron 3 to iron 2 before it can be absorbed. And at this site now, for it to be incorporated into transferrin, it must be converted back to iron 3. And the enzyme that does that here at the duodenal enterocyte is called hepstine. You also have this type of enzyme in plasma, and in plasma it is called celluloplasmin. You also have it in placenta. In placenta it is called cyclopen. So here in the walls of duodenal enterocyte, you have the hepstine that helps to convert the iron 2, absorbed iron 2 that had passed through the ferroportin back to iron 3 before it can be incorporated into the plasma transferrin. Once it is incorporated inside plasma transferrin in the blood, it is now passed through the blood vessels and is either carried to the liver for storage in the ferritin that are in the liver, or is taken to bone marrow where it is involved in erythropoiesis, or is taken to other sites of utilization, other sites where iron is being utilized. Now, iron, when it is incorporated into transferrin, remember it is incorporated into transferrin in form of iron 3. This transferrin will now carry it to cells that actually need iron through the blood vessels to cells where it is needed, bone marrow, liver, and other sites where iron is needed. Transferrin cannot take iron into the cells like bone marrow and all the rest of them, or red blood cells, for example, that need iron, mitochondria that need iron, or in form of formation of hemoglobin. Transferrin cannot take iron inside those things. They need what we call transferrin receptor. These transferrin receptors are found on the surfaces of red cells, on surfaces of cells that need iron. They accept this iron from transferrin and then internalize it into the cells. So the major function of transport proteins, transferrin, is to move iron from wherever it enters, whenever it enters the plasma, that is through the intestinal villi, splenic sinusoid, to erythroblasts of the marrow, and to other sites of utilization. That is the function of the transferrin, is to move iron through plasma to all the places where iron is needed, to do its work, the points where iron is being utilized. Now, dipheric transferrin, that is iron that's bound in form of iron 3 to transferrin, binds to the transferrin receptor. Remember, I said these transferrin receptors are found on the surfaces of the cells that actually need iron, because transferrin on its own cannot take iron inside these cells. Point of utilization, like for example, hemoglobin is found inside the red blood cell, and hemoglobin, red cells, young hemoglobin, young red cells that are being produced in form of erythropoiesis, need iron for them to produce hemoglobin. And this ion needs to get inside the red cell for it to be incorporated into the heme. And transferrin, the work of transferrin is to carry ion through the plasma to the sites, to the surfaces of the red cells. It may not be able to internalize the ion inside the red cell. So it needs what we call transferrin receptors that are found on the cell surfaces. So this transferrin bound with ion binds to this transferrin receptor on the cell surface. And the transferrin receptor complex forms clusters in the piece on the cell surface. The complex is then internalized by endocytosis. When this transferrin that is carrying ion meets the transferrin receptor on the surface of the red cells, they form a complex. When they form a complex, the complex is then internalized by a process called endocytosis, internalized within the cell by a process called endocytosis. Within the cytosol, that is the cytoplasm of the cell, the transferrin receptor complex is in a platinum-coated vesicle. This vesicle fused with the endosomes, in which occur acidification and the release of ion from the transferrin. So within the cytosol, the acidification and then release of this ion from transferrin occurs. When ion is released within the cytoplasm, the released ion is now used in production of hemoglobin, while the apotransferrin receptor complex returns back to the cell membrane, where at the neutral pH, the apotransferrin is transferring without ion now. It's released back into the plasma, and it re-enters into the plasma to take up more ion, and then the process repeats itself again. So when ion is absorbed through the divalent metal transporter 1 for non-heme ion, and then heme transporter for heme ion in form of ion 2. Remember, ion 3 must be reduced to ion 2 before it's absorbed. And then when it enters into the duodenal cells and move at the other side of the enterocyte through the ferroportin, well, as we described here, it must be reoxidized back to ion 2 and incorporated into the transferrin. The work of transferrin is now to carry this absorbed ion to where it is being utilized. And transferrin cannot on its own move ion into the cells where it is needed. Transferrin needs what we call transferrin receptor that is found on the surface of the red cells and other cells that need ion. The transferrin receptor form complex with transferrin that's carrying ion, and this is now internalized through a process called endocytosis. Within the cytoplasm of the cell, the acidification process helps in releasing ion from this complex, and then the complex is returned back. And at the surface of the blood cell, transferrin will release from the transferrin receptor, and transferrin goes back into the plasma to repeat the process all over again. Now, ion in the erythroblast, then within the erythroblast, within the young red cell that is developing red blood cell, once within the developing erythroblast, ion must be transported to mitochondria to be, remember, in hemoglobin synthesis occurs largely in the mitochondria and then in the cytoplasm of the cell. Once within the developing erythroblast, ion must be transported to mitochondria to be incorporated into heme or taken up by the ferritin within the siderosomes. Within the vesicle, divalent metal transporter that is also involved in transporting ion effects the release of ion 3 into the cytosol, where it is taken up by mitochondria for heme synthesis. Within the mitochondria, ion is inserted into the protoporphyrin ring by heme synthase, fellow Charlie test. So when ion is made available, the ion is now incorporated into the protoporphyrin ring that had been formed earlier for it to form heme part of hemoglobin. So ion must be taken into the developing erythroblast by transferrin receptor complex. Once it is internalized through the process of endocytosis, the iron is released within the cytosol, and then the complex return back, and the iron is incorporated into the hemoglobin in the into the heme in the mitochondria. That's protoporphyrin ring in the mitochondria to form heme. When we are discussing absorption, I said that factors that stabilizes iron 3 reduces iron absorption. Remember, for non-heme iron, iron 3 must be reduced to iron 2. So if you have things in your diet that makes iron 3 to remain as iron 3, that means iron may not be absorbed, because before non-heme iron must be absorbed, but must be absorbed, they must be reduced to iron 2. So things that stabilizes iron 3 decreases iron absorption, especially non-heme iron. Why things that stabilizes iron 2 increases iron absorption? So factors influencing dietary iron absorption. For heme iron absorption, iron status of an individual can influence. If somebody needs more iron, there's a tendency for increased heme iron. When you eat a lot of meat and poultry products and fish, they can increase absorption of heme iron from your diet, can occur because your system needs more iron. Pregnant women, those that are anemic. Then amount of dietary heme iron, especially as meat, the amount of dietary heme iron that you have in your food can also determine the amount of heme iron that can be absorbed. Then calcium reduces iron absorption. Form complexes with iron and reduces iron absorption. Food preparation, the time of cooking, temperature. Too much cooking of food also reduces its absorption, and then the temperature of the food. Then we have non-heme iron. Non-heme iron status of individual can also affect iron absorption. Remember, we said active erythropoiesis, that is somebody that needs active erythropoiesis increases absorption, while iron overload reduces iron absorption. The amount of potentially available non-heme iron, that is the amount of iron you have in your food, also will determine the amount that should be absorbed. And then balance between enhancing and inhibiting factors. If we have factor that stabilizes iron 3 in your food, those things will inhibit non-heme iron absorption. But you have factor that stabilizes iron 2 in your diet, it will also increase absorption of non-heme iron, increase absorption of non-heme iron. Now, let's look at enhancing factors, factors that enhance absorption of non-heme iron. Like we have enhancing factor like reducing agents like ascorbic acid. Ascorbic acid that you found in some juice, fruits, potatoes, and certain vegetables helps in enhancing iron absorption. You have hydroquinone, lactate, pyruvate, succinate, fructose, cysteine, and sorbitol. All these things help in enhancing factor that enhance iron absorption. That having iron in form of heme, like meat, chicken, fish, seafood, increases iron absorption because heme iron are absorbed more readily than non-heme iron. Fermented vegetable like fermented soy sauce and everything, and also increases here iron absorption. So heme iron is absorbed more readily than non-heme iron. Remember, I said that non-heme iron must be reduced to iron 2 before they can be absorbed. You have inhibiting factor, things that can inhibit non-heme iron from being absorbed. Phytates and other inositol phosphates, example, brand products, bread made from high extraction flour. You have oats, especially unpolished rice, pasta products, coconuts, soybeans, and peas. Then we have iron-binding phenolic compounds like tea, coffee, cocoa, certain spices, certain vegetables, and most red wines. And then we have calcium. That is why most times it is not good when you are on iron supplementation. It's not good to take it together with calcium supplements because calcium retards iron absorption. Calcium retards iron absorption. So when you're on iron supplement, the best thing to do is either you take iron in the morning and you take your calcium in the evening. it is wrong to take both of them at the same time because calcium affects iron absorption negatively. If you take it with calcium, you might be taking your iron supplement, but you may not be getting anything from it. You may not be getting your desired results from it. Iron cycle in humans, once an atom of iron enters the body, either through diet, food that we eat, either as heme iron and non-heme iron, and it is absorbed through the divalent metal transporter 1 when it is reduced to iron 2 across the luminal wall of the duodenal enterocyte and then carried across the enterocyte walls by ferroportin and changed back from iron 2 to iron 3 by Hemostasis. It is incorporated into plasma transferrin and carried inside the plasma, extracellular fluid to developing erythroblasts and phagocytic macrophages. There, when it is used in producing red blood cells, and these red blood cells circulate within the blood for at least 4 months, that is within a period of 120 days. It then travels to phagocytes of the reticuloendothelial system, where senescent erythrocytes are engulfed and destroyed by macrophages. And when this is done, the iron part of the erythrocyte are recycled and reused again, taken back into the pool again for the whole process to go over again. Again, about 10% of newly formed erythrocyte normally is destroyed within the bone marrow, and its iron is released and reused again. This is called ineffective erythropoiesis. The iron from ineffective erythropoiesis is also taken back into the common pool and reused there again. Storage sites like ferritin releases a small portion of iron, as the body needs. And then it can also have a small proportion being lost in urine, sweat, feces, blood, and the equivalent small amount of iron is also reabsorbed through the intestinal tract. This is kind of iron cycle. As iron is being absorbed and used in producing red cells, enzyme that is being needed, the matured red cells being destroyed, and the iron that is from there is being reused back into the pool and is also reused there. Again, there is a balance between the amount of iron that is absorbed and the iron content of the body. And again, because the body does not have a physiologic means of losing iron, a small proportion of iron is lost through the shedding of dead cells of the skin or through sweat, and a small amount is lost through urine and feces. Unless we have acute blood loss where you can lose substantial amounts of iron, or women who menstruate can also lose a higher degree of iron compared to male counterparts. Looking at iron hemostasis, intestinal absorption of iron. From there, you have iron is moved into the plasma transferrin. 2 things happen there. It's either taken to liver for storage, or it's taken to marrow erythroid for formation of red cells. And then from there, you have the circulating red blood cells. And then, or it's either taken to skin, gut, endometrium, and then stored as mucosal ferritin, and subsequently shed away when the shedding of skin cells is done. And that amounts to about shedding of blood, shedding of dead cells about 1 to 2 milligrams per day. And then circulating erythrocytes, about 120 days, can migrate through the mononuclear phagocyte stores when cells are destroyed and the iron content is reutilized, taken back into the common pool, and then join with the intestinal, the one that absorbed from the intestine and recirculated all over again. What have we said? In this lecture, we have been able to say that iron is absorbed at the proximal duodenum, and we have proteins that are involved in iron absorption. The chief among them is what we call divalent metal transporter 1 that carries iron, transports iron across the duodenal wall. But before this protein absorbs iron, helps in absorbing iron, iron must be reduced. The iron in our diet occurs in two forms, heme iron and the non-heme iron. The heme iron can be transported directly through the heme transporter, while the non-heme ion occurs as iron 3 and must be reduced to iron 2 before they can be absorbed. And in reducing it to iron 2, we have enzymes like cytochrome B on the luminal wall of the duodenal enterocyte to reduce iron from iron 3 to iron 2. When this is done, the iron is reabsorbed. And for it to move into the circulation, they move into the circulation through the duodenal enterocyte through a channel called ferroportin. And this ferroportin allows iron 2 to pass through it. And when this is done, the iron 2 must be converted back to iron 3 before it can be incorporated into transference, which is the protein that transports iron in the plasma. The enzyme that helps in converting iron 2 back to iron 3 is hemostasis. It is found on the duodenal wall, and it can also be found in the plasma as ceruloplasmin and in the placenta as cyclopen. Now, when it is converted back to iron 3, it is now incorporated into transferrin. Transferrin now carries this absorbed iron either to liver where it is stored as ferritin, or to erythroid cell, that's bone marrow, where it is needed to produce red cells and other cells that actually need iron. But transferrin cannot take iron inside the red blood cell. There are what we call transferrin receptors that are on the surface of these cells that form a complex with this transferrin so that this iron can be internalized inside the cell where it can be utilized to produce hemoglobin. So the transferrin receptor on the surface of red cells bound to the transferrin that carry iron and form a complex, and it is not internalized through a process called endocytosis, and it's taken inside within the cell, the erythroblast. Within the cytosol, the iron is released and taken to mitochondria, where it is incorporated into the protoporphyrin ring to form heme. While the transferrin receptor and transferrin complex is taken back to the surface of the cell, where transferrin is released back into plasma, and the cycle is repeated there again. So when this is done, there's a tight regulation of processes that are involved in iron absorption. Like I said, there are substances that enhance absorption of iron from our diet, like reducing substances that helps to maintain iron in iron 2 states. They enhances iron absorption, but those substances that help to maintain iron in iron 3 states reduces iron absorption. They reduces iron absorption. Those things that stabilizes iron and keep iron in iron 2 enhances iron absorption, while those that keeps non-heme iron at iron 3 state reduces iron absorption. While non-heme iron is readily absorbed more than non-heme iron. Heme iron is readily absorbed more than non-heme iron. So we are going to look at iron. In the next lecture, we are going to be looking at regulation of iron absorption. How is iron absorption regulated? How is iron absorption regulated? That is the next lecture that we are going to have. I hope that you have learned one or two things from this lecture. Keep a date with us in the next class. God bless you. Thank you for